do you have a greater appreciation for, I guess, shoes now? And I can imagine all the engineering that most people, probably right over their head, they just think it's another pair of shoes and it's either comfortable or it's not.
Yeah, absolutely. It's really cool. So much of it is driven by materials.
Hello, my name is Adal Said, and thank you for joining us today in April's ASM Ontario talk. Thank you for being here today. So I also always want to thank the other executives who are here to help host these talks. Jordan, Mernali, Danielle, as well as some of the other members of the executive team who are more in the attending list and not in the panelist. I also want to thank all our Canadian chapters as well as chapters in the US, Mexico, India, UK that helped promote these events and start bringing their members in. We really appreciate your attendance, as well as all the local universities who helped advertise the talks and try to bring in as many students as possible. So we're constantly looking for students to be representatives at our local institutions. So if you are interested in being that representative for ASM Ontario, please reach out to us. I could be reached at chair@asmontario.org. Or you could just search my name in Google too. You could contact me through my university email. So to hear about future events, please sign up to our newsletter at asmontario.org. And there you have links also to our previous talks. So you can see recordings of our previous talks that we had before last year, as well as the ones that we've just had last month or 2 months prior. Our question master is actually busy with their capstone project. It's due tomorrow for them. And so they couldn't be here today. So I've been taking over as the question master for today. So if you have any questions, please send them to me directly during the chat, and I'm going to curate them and I can give them to our speaker. Now I want to pass it on to Jordan, who will help introduce our speaker.
Alrighty. Good afternoon, everybody. Hope everyone's doing well. So as Abdallah just said, I'm Jordan Wilhelm, the current chair of the ASM Canada Council. Well, ironically, when I gave the intro to last month's talk, I started with the idiom, try putting yourself in someone else's shoes. Now today, the saying takes on a more literal meaning. So shoes have been around for thousands of years. They have survived the test of time and have evolved from a simple piece of leather hide to a $350 billion industry. Now, through a little bit of research, by comparison, steel is only estimated at $320 billion. So it's massive. Now, when you think of shoes, their basic purpose can be simplified down to something so simple, to protect your feet. But footwear has become a necessity in fashion, safety apparel, keeping you comfortable in any weather conditions, and of course, athletics. Shoes have been engineered to help humans reach our peak performance, whether it's running fast, jumping high, or creating fluidity in one's motion. If you're working from home today, I hope you're wearing your comfiest pair of athletic slippers to help appreciate our athletics footwear expert. Please welcome Ransburg Professor of Materials Engineering at Purdue University, Professor Nikhilesh Chawla.
Well, thank you very much, Jordan. Appreciate the introduction, and I want to thank Abdallah and Mernali and Danielle and the crew at ASM Ontario for inviting me to give this talk. And so I'm sharing my screen here, and hopefully you all can see this.
Yeah, I can see it. I can see now it's going.
So what I'd like to talk about today is really some work we've been doing over the last several years, primarily with Adidas, and looking at something called Boost Foam, which I'll be talking to you about and telling you about today. And so the title of my talk is Materials Characterization and Mechanical Properties of High Performance Athletic Footwear. And I want to thank one of my really key former PhD students, Arun Sundar, who did this work was really a majority of his PhD dissertation, and Arun is now working at Thermo Fisher Scientific. So maybe just to talk a little bit about what were we really looking for when we're designing? First and foremost, of course, it has to be lightweight, right? So we need to be able to save energy and in order to get better performance. And nowadays, shoes, both running shoes, and then you talk about soccer cleats, these materials, that they're using materials and technology to really make athletes faster, more agile, and stronger, and get that that advantage in whatever pursuit that they're going for. So saving energy is key to better performance. Flexibility, of course, is key. We need to have this kind of soft rubber-like behavior, and you also need to get some rigidity as well. You don't want something that's flapping around. Has to be able to be used in all kinds of seasons and all kinds of climate zones. So all the way from the winter in Ontario and Indiana to summers in Arizona and in other deserts around the world. So it has to be able to withstand those very large temperature gradients. Processability is big. So the designs are becoming a lot more complex. You need to have shorter cycle times. Customers are becoming more and more demanding and selective. And the competition is certainly increasing. And if you go to the websites of Adidas or Nike, you can now actually make your own shoes. You can design your own shoes, and you can get those hopefully, in a matter of weeks. And I have two boys who play soccer. I'm a soccer coach, and I know that having really funky looking nice cleats with their names and their uniform numbers on the shoes are the rage these days. So that's a big thing. Let me talk a little bit about the history of high performance foams. And this is a slide that's courtesy of Adidas. And so if you look back at the sort of really older Adidas shoes or even Nike shoes, they look something like this. They were all nice and white with 3 stripes. And nowadays you have a variety of different kinds of materials. You have these, the midsole compound, of course you have the rubber, and then you also have technology that goes into this. So things like the My Coach. This is a little chip that goes into your sole, your shoe sole, and if you're a soccer player, for example, it can keep track of how much you've run, what your average, you've struck the soccer ball, and things like that. So there's a lot of data that can be achieved from this. Okay. And so one of the early sort of proponents for foams in materials is something called Adiprene. Okay, and essentially that was one of the early versions. And since then, they have moved to something a lot better. So here's actually a little video comparing the EVA, which was the classic sort of foam material, with the Boost foam, which is really the topic of today's presentation. And we're going to show you a video of just dropping these steel balls on concrete and EVA and Boost. And you can see that that ball that's bouncing on the Boost is continuing to bounce well after the other balls have stopped bouncing. So you can see that the Boost in this very simple example gives you very nice amount of flexibility and rebound to your foot, whether you're running a marathon or whether you're walking in your neighborhood. It's going to give you that comfort. So what is Boost? And so Boost was actually introduced back, I think, in 2013 or so. And it's really a composite, tiny little holes, and it's really a hierarchical, porous foam structure. Okay, and I'll show you exactly what that structure looks like. Okay, so you have these little beads, and all of these beads here are actually shown in a ball, but essentially these beads are used with a steam chest molding process, and you actually then form this sole of your shoe. It looks kind of cool as well. It looks like you have all these tiny little beads in there, but they're actually all fused together into the plate of this boost foam. Okay, and this slide actually shows you a discrete element modeling, CFD kind of simulation, showing how these beads are actually flowed into the sole to actually make the footwear, and essentially they flow all the way through, and then eventually they apply some temperature and some steam, and you get the fusion between these beads. So in reality, what boost foam is a very unique multi-scale hierarchical foam structure. It essentially has very nice attractive properties, energy absorption, has high strength per unit weight. The length scale of this, on a macro scale, you have all these beads together, and then we need to look at the individual beads and what the structure of those is. And then we can even go down to the ligament level and the cell level, which I'm going to talk about today using techniques like X-ray microtomography. And if you wanted to go even further down to the nanoscale, you could get into the overall polymer chains and the chain structure of this particular polymer. So really the objective when we started this work was, and Adidas had come to us and said, we have this really interesting material. We really don't quite understand the microstructure property relationships in this foam. And so that was really the guiding force here, is how does cell size and morphology affect flexibility? How does it affect compressibility? How does cell distribution, how does ligament size, and all of these structural effects, how does the 3D structure of the individual beads as well as the overall plate affect mechanical properties? And so we set out to do these in situ experiments to really look at how the overall cell structure is affecting the compressibility and the flexibility of the materials. And this was actually done using the X-ray microtomography under compression. And I will describe some of those experiments. So how are these beads made? So we have these pre-expanded beads, and then essentially these are put into this kind of an autoclave kind of thing, and then you inject this gas, and the pre-expanded beads then actually get foamed. So if you think about a balloon that initially does not have any air, you fill up that balloon with air, so you foam these beads, and then you essentially get this the cellular structure, the porous structure. Okay, so this is actually now an SCM image of the cross section of the bead, and you can see here. So this is the bottom portion of the image. You have the core of the bead. So the first thing that you can tell here is that this is a graded structure. It's much denser on the outside than it is on the inside, and you have much larger cells on the interior of the bead as well. And this particular foam, if you notice that one of your sponsors was LICO, and so sample preparation, of course, is so important. This one was actually made by ultrafine sectioning technique, a microtoming technique, where we have this very sharp blade that can cut through and give you a nice surface quality without having to polish the sample. Okay, so this is actually here, you can see on the surface region, you have much finer cell size distribution, and then you have the transition region here, and in the center and in the core, you can actually see much larger cell size. Okay, so now we can transition a little bit to the overall plate distribution. So if we start to get these beads together and then we fuse them, and I want to actually show you these images on a little bit larger scale. So here's actually a nice image of a cross section of the plate from the shoe beads that have been fused together. And you see this darker gray region where you have very nice bonding. And in some regions you have a little bit of defects, but overall very nice bonding here. And you can see that the graded structure actually gets, the cell size gets smaller and smaller as we get to the inter-particle or inter-bead boundaries. And so you can start to see now how the overall packing of the beads is going to affect the load transfer and how the material behaves under compression. Here's another example of that fused region. So you have a very nice thin region, which is giving you the adhesion between the beads. And in some situations, you do see some delaminated regions, okay? And these, over time, are going to initiate some interfacial cracks, most likely. So, so now the question is, and what I showed you so far were all sort of cross-section SCM images, destructive images. And we wanted to find a way where we could look at the microstructure of the material in 3 dimensions, non-destructively. And we also wanted to be able to apply a stimulus. And in this case, the changes in the structure while the material is being compressed. And so you're all familiar with the nice tetrahedron here, where we look at structure and processing and properties. And what I show at the bottom here is how we can use these wonderful techniques that we have in the year 2021 to study these properties, and that can go all the way from light microscopy to helium ion microscopy, and we use X-ray microscopy, as I'm going to show you, and of course SEM and focus ion beam microscopy. I mentioned here about 4D, and really what it really means is we're looking at the 3D structure, and the 4th D is really time. And you know, 4D is not unique just to material science. There's 4D movie theaters, there's 4D printing, there's 4D smart materials, and there's even things like 4D autofocus technology. And this is probably most analogous to what we're doing with tomography, because here with the autofocus, you're actually looking at time-evolved images, and in this case, they're using focusing technology to predict what the object is going to do. So can we match lengths like fracture or fatigue or creep? You know, these things span multiple length scales, and we can start to link now these particular properties to individual imaging techniques. So we're going to be looking primarily at X-ray microscopy, X-ray microtomography. We can go all the way from the millimeter to submicron regime. And I'll just tell you a little bit about our center, which started at Arizona State University prior to my moving here to Purdue about a year ago. And we had a kickoff meeting about 4 years ago, and these are our wonderful sponsors who fund our research. And here at Purdue, we have this X-ray microtomography capability where we can do imaging, and this is a cross-section of the boost foam. But for metallic materials, which I'm not talking about today, we can do things like 3D crystallographic, 3D EBSD kind of measurements. And we also have capability to do nano indentation all the way up to 800C and down to minus 120C, as well as things in the liquid cell. And this is open to all internal, external users. So if anybody is interested in using the facility, please feel free to email me. Okay, so let's talk a little bit about X-ray microtomography, and it's a technique that I 10 or 20 years, and it's been primarily used for imaging. But as I mentioned, you can also use it to get 3D crystallographic information. And it's ideally suited, because it's non-destructive, to the kinds of 4D experiments that we wanted to do. Okay, and so if we want to do tomography, what exactly does it mean? And so I'll just put a little cartoon here So if we wanted to look at the 3D structure of a light bulb, we would actually shine some X-rays through that light bulb, and we would get two-dimensional projections. And then we would actually turn that light bulb at angular increments of 1/8 of a degree or 1 degree. And essentially, you would get a two-dimensional projection. When you get all of those two-dimensional projections, you can use what's called a filtered back projection algorithm, which is essentially an inverse Fourier transform of that data, and you can get your 3D microstructure. Okay, so the data acquisition part of this and the reconstruction part of this is certainly important, but there's a lot of things that we want to do with this data downstream, and that requires a fair amount of image analysis, image segmentation, visualization of these data sets, and then, of course, statistical quantification to the image analysis to try to actually understand what the mechanisms of deformation are taking place. And that could be, in this case, changes in cell size, changes in ligament size, ligament diameter. In fracture cases, you would be looking at things like crack density and things of that sort. Okay, and one of the other things that you can do is you can take this microstructure and incorporate it into a finite element model, and you could actually use the initial microstructure to simulate what the material behavior is, and then of course you have the 4D experiments to compare it side by side. So the first part of my talk, I want to talk some experiments we did on in situ compression in the lab. So this is a lab scale X-ray source. And in the second part, I'm going to talk to you about some experiments also in situ compression, but using a synchrotron source at the Argonne National Laboratory. And there are different reasons for doing the synchrotron experiments. Those experiments are more dynamic. So this is actually an image of the Versa 520. Here at Purdue, we have Versa 620. You have the X-ray source here, and there are several filters. So it's a polychromatic beam, and the or low end or both of the X-rays coming in. Your sample is going to be sitting here, and it's going to be rotating, and we're taking projections at each of those angular increments. And then you can also actually get some magnification in this part here. There's a scintillator screen that converts the X-rays to visible light, and then there's a CCD camera that captures the images. Okay, and of course this is done with the doors closed. We take care of safety. Take safety very, very seriously. So that's the X-ray part, but we need to be able to now compress the foam and be able to keep it at a particular strain. So Arun actually devised this very neat little in situ loading fixture where your sample goes in here, and you have these rotation of these screws that impart a downward displacement. And you need to have this PMMA sleeve to give some robustness to the cell. And that PMMA material is X-ray transparent. Okay, and you have a load cell down here. And so this whole thing is going to have to be able to rotate 180 or even 360 degrees during the X-ray tomography scan. Okay, so this is actually an image of that. You can actually see this is one of the beads between two of the compression platens there, and the compression direction is in this direction. Okay, so one of the first things we wanted to look at is, okay, are the X-rays going to be affecting the mechanical properties of the foam? And so we did multiple cycles, and we exposed these to the X-rays multiple times, and these are load versus displacement curves under compression, and you can see that they stay pretty much the same. And these initial experiments are on individual boost beads. Okay, and so here I want to show you actually one of the data sets So this is one of the beads. Okay, so you have the top platen here, bottom platen here. They're actually a little bit oblong. Start to see some of the cells there. Okay, so this is actually before the experiment. And then I'm going to play you another video here after we sort of squished that bead to about 50% strain. And then you can actually take an X-ray scan of that cellular structure. Okay, so now you can start to study the overall. And remember that I mentioned that these beads have a graded structure and they're denser on the outside and much coarser with thicker ligaments on the inside. Okay, and so here you can start to see that most of the action is actually taking place on the periphery of that bead, and most of the densification is happening there. Okay, and so there's very little strain, very little deformation going on in the interior of the bead. Okay, that's what we call region 2. From the outside of the bead, you see most of the deformation. All right, and here I'm going to play you a little time-lapse video, and the percentage of compressive strain is actually on the top left-hand corner of the image. So here it is under compression, and you can start to see, again, most of the damage, most of the densification is taking place on the outside. And one of the other things you see is this very interesting kind of crinkling and ligament buckling in some of these. So we started to look at this in a little bit more detail. So here's another example of how bead cell shape and orientation to the compression axis also plays a role. So whether this is at an angle or if it's perfectly parallel. Important, and before we could do any of the statistical quantification, we had to actually go through a lot of this image processing. So we use these kind of non-local means filters, and then we do a grayscale threshold. And I won't go through a lot of this, but essentially there's a lot of image analysis that goes into actually trying to make sure that you're actually capturing all the ligaments and that they're not actually joined through some kind of an artifact of the imaging. Okay, and so part of this is actually what's called a region grow algorithm. And so essentially we're looking for pixels or voxels that are of similar grayscale value, and that will continue until it's almost like walking in a cave until you hit the cave wall. And so that's what we're looking for. So when we hit the cell wall, the grayscale changes, we stop the segmentation of the cell. So this is actually an example of that. And then what you're able to get is overall imaging of the individual cells in the foam. Okay, and so here's actually each of those nicely colored cells is a different cell, and you can now start to look at the and one of the really cool things is that these cells are not perfectly smooth. So they almost look like we filled up a balloon and then we took a little bit of air out, and some of that balloon, each of these, if you think of each of these as a balloon, shrinks down. And so we get these kinds of cells with a fair amount of curvature. And this really has to do with the process by which the steam, the blowing process. So once they foam the beads and essentially you cool it down, there's going to be a contraction. And so each of these individual cells is going to go through this slight, crinkling process during the cooling part of the processing. Okay, so that's what the void actually looks like. All right, and so now with the 4D part, you can track individual beads to actually see what they look like as a function of strain. So I'm going to pick 3 different cells here, which are shown with the blue, the red, and the yellow stars. And we're going to follow those individual beads as a function of strain. Okay, and so essentially what you see is that you have initially nicely shaped beads, and over time they start to crinkle down. The individual cell walls actually go through a very microscopic process of buckling. So you start with almost these straight ligaments, and eventually they become snake-shaped, and they end up buckling, in a very localized way. And these are the kinds of things that you can really capture using the X-ray tomography. Okay, and so here you can start to see that the stresses close to the surface actually end up being higher and higher, okay, because we're getting through the densification. The middle of the cell is actually still in the elastic regime. And then now you start to see that we're sort of deciphering some of the secrets as to why Boost is such an amazing material, because really only the outside of the individual cells are deforming and densifying. So this is actually change in volume, just to show you that quantitatively. So this is the change in volume on the surface in the blue. And then in the central core, you can see that all of those cells are actually not changing in volume very much. Most of the action is taking place within 500 microns or so of the surface. Okay, and this is as a function of strain. So as you start to compress and get more and more strain, you can center of the beads, and you have now this gradient in strain essentially, and it progresses towards the central core. So I showed you changes in volume of individual cells, and so now we actually went through a process where we wanted to get localized strains. And for this, we use digital volume correlation technique, which is very similar to digital image correlation, which is a 2D technique. DVC, or digital volume correlation, is a 3D technique. And so here we're going to be using these cells and cell walls and the centroids of the cells as markers, and using the displacements of those markers to get displacement fields. When we get displacement fields, we can calculate the strains. So this is just a schematic of that. And so now you can start to see the overall local distribution of strain. This is at an applied strain of 5%. And as you get to larger strains, you can see a lot more of the strains in some of the thinner ligaments. And that's a precursor to that buckling phenomenon that I alluded to. Okay, this is actually just a plot of that compressive strain from DVC and the applied strain. So again, most of the strain is on the surface near the actual platens as compared to the central region of the core. So okay, I'll just show you another nice image of this compression and the effect of the wrinkling for each of these individual cells. Okay, and the second part, I want to talk now just about some of the experiments we did at the synchrotron facility. And so here there were a couple of reasons for doing this. One, we wanted to look at graded cells versus homogeneous foams, so beads with homogeneous structure, as opposed to the graded structure that that I showed you with the Boost. And we were expecting that the homogeneous cells, which had a higher density, would also be stiffer. And I'm going to show you that that was actually not the case, which is a very interesting finding, that we actually had higher stiffness with the graded cells than we did with the homogeneous one. And that, again, has to do with the graded structure. Okay, so here's the motivation for doing the synchrotron experiments. And so if you look at this plot in the lab, the scan times are fairly large, so 10 hours, 14 hours. But more importantly, when we compress the beads, you have a stress relaxation effect. Okay, and so here is actually the stress relaxation behavior, and it starts to taper off after about 5 hours, but it really doesn't get to be constant until about 20 hours. So you can imagine that you have to let the material relax for about 20 hours, and then you have to scan for another 10 hours, and that's really time consuming. So at the synchrotron, you have X-ray fluxes that are 5 to 7 orders of magnitude larger than what you have in the lab source, so 10 to the 12 photons per second. And you can also get a very monochromatic beam as opposed to the conical beam, that polychromatic beam that I talked about with the lab scale. So we went out to the Argonne National Laboratory, and this is what the synchrotron ring looks like. And essentially, I think the circumference is probably about 1.5 miles to 2 miles. And in each of these areas, you have individual beam lines where you can do different kinds of experiments. And we're using a beam line called 2-BM. And we have these electrons that are essentially go at very, very high speeds. And we have these undulator magnets that essentially they go back and forth, and then essentially you get these X-rays that are transmitted. So you get very large, very high brightness flux of X-rays. Then you can use what's called this monochromator, and essentially then it hits your sample. And so the point is that you can now do, going back to this slide here, instead of waiting 20 hours for stress relaxation and another 10 hours of scanning time, you can do these experiments on the fly in real time while the foam is being loaded without having to pause the experiment. Okay, so these were the experiments that we did at the synchrotron. We're actually scanning for 360 degrees of 2 seconds. You have 0.5 millisecond exposure, and you can actually do an actual compression experiment in about 40 minutes. Okay, now, so one of the keys with the synchrotron facility, and even with the lab scale facility, but primarily the synchrotron facility, is that this in situ compression jig has to weigh less than 2 kilograms. It has to be able to rotate, of course, 180, 360 degrees. And the X-rays have to be able to pass through the sample and come out the other end without being obstructed. So, and in this way, the X-ray beam has to go through. So again, we use a capped-on tube. You have the compression platens here. This is the loading motor, and you have a bunch of these screws. For anybody who does mechanical testing, you know that axial and concentric misalignment is very important. So we can actually use the images to see how much misalignment we have, and we can correct for that because we have three-dimensional imaging capability. And so so this was actually one of the scariest parts of the trip, is the image on the right. And the first experiment we did, we noticed that the beads were in black. And so, my goodness, we're zapping the beads with the X-rays from the synchrotron. We have a huge amount of flux, and we're essentially frying the beads. And it turned out that we had accidentally left the shutter on, even when we were not scanning the sample, and that extra exposure to the high flux of X-rays was causing damage. And when we fixed the shutter issue, we did the experiments again with low displacement to make sure that the X-rays were not damaging and things were fine. So that's what we call a heart attack moment. So here's actually some of the data sets that we got from stitching different volumes. So you can imagine that we're essentially scanning in a helical way and stitching the different volumes and getting the entire data set. And one of the things that you can do with this data, I'll just skip through some of the imaging here, is you can calculate things like Poisson ratio. Okay, and it turns out that the Poisson ratio, which is the ratio of the lateral to the axial strains, can give you a lot of information of what's going on in the bead. So here are actually 3 different beads that we looked at, and this is for the homogeneous bead, so graded versus homogeneous. And now you can start to see volumetric strain versus compressive strain. Okay, and each of these numbers here is the predicted Poisson ratio for those curves. So if it were 0.5, you should see it closer to this curve. If it was close to 0, it should be close to this curve. And you can see that most of these foams are somewhere in the range of 0.25 to 0.33 or so. And we compare this to the lab scale data, and we actually have good agreement there. So that was good news. Okay, and so what's happening here again is you have this compressive stresses going on. You have tensile stresses going sort of radially. The thicker ligaments are the ones bearing the load. And again, most of that deformation is taking place towards the outside, particularly in the graded bead. Okay, and so you can actually see that here. So one of the interesting findings, and I will kind of end with this, is that even though you have a lower density bead, okay, so essentially in this axis, the x-axis, the graded bead is stiffer for a given density compared to the homogeneous bead. The reason is that the outside of that bead is actually taking up most of the load, giving a densification, and locally giving you a more stiffer response. So I'll just go through this very quickly. Again, we looked at localized microstructural impacts on compression, use digital volume correlation to get local strain maps and how heterogeneities were affecting the onset of these local buckling events. And then in the last part, I talked more about the synchrotron experiments, which were the dynamic experiments, where we could actually measure things like Poisson ratio and link those to the microstructure. So I'll end there, and I would like to thank again Arun Sundar, who is the PhD student who did all of this work, who's currently at Thermo Fisher. It's recently graduated. Collaborators at Adidas. So Yasmin Max and Chris Holmes are in Germany, and Mark Henderson is here in Portland. I should mention one of the perks of working on this project is that we got some discounts and some free footwear of Adidas Boost foam. So, and if you have any more, if you would like to get more details on this work, you can look at these 2 pages. But if you're interested, I can send you a copy. And the one on the bottom actually was published recently in Journal Material Science. So I will end there, and hopefully my talk did not cut out anymore, and I'm happy to answer any questions.
Yeah, no, look great from my end. So I'm going to let Danielle take it away, though. So.
Yeah, thank you, Professor. I really appreciate you taking the time out of your schedule to give us your talk. I definitely learned something new, and now I think I'm going to have to go buy myself a new pair of Adidas running shoes to replace the other brand ones that I have, because I don't think they have the Boost foam on them. Actually, no, I know they don't, because they're not Adidas. So I got to get myself a new pair of running shoes. Again, if you have any further questions, please send them to Abdallah in the chat. We understand that we're scheduled to talk for 1 hour, so if you need to get going, you can. This is just our thank yous. So thank you all for attending. While we have you here, I just wanted to let you know about our next event. We have another ASM world-class speaker scheduled for May. It's May 5th at 1 p.m. Eastern time. This one will be on the materials for nuclear fission. And so the talk will include an introduction to nuclear fission and also talk about the materials that are used at the very high temperatures used. I wrote down in my notes extremely high temperatures. I think it was up to the millions of degrees Celsius. You can find all the information on our website, asmontario.org. There you can sign up for the talk and also sign up for our emailing list, which will give you reminders about the talk and tell you how to register. So thank you all for attending. I'm going to pass things over to Abdallah, who will get our question and answer period started.
Thank you, Professor Nick, for the neat talk. Yeah, those videos and that kind of characterization work was really amazing. I know it's really difficult to get some kind of imaging and tomoscopy and tomography, especially for polymeric materials at that scale, and dealing with the different size gradients. That's always kind of like a researcher's nightmare, dealing with so many different time or length scales at once. That's where it gets tricky. So we have some questions, if you're willing to kind of go through some of them.
Of course.
Yeah. So there's a lot of students who tuned in, and I think that I tried to kind of combine two of them because they were in the same area. I think they were very, they want to kind of enter the career in materials in athletic wear or wearables or kind of footwear. And so, and I saw, it looks like
Arun Sundar.
Arun, yeah, he had to have a very good grasp of all the tools he's using.
Yes.
So the question I wanted to kind of, I'm trying to craft it seems like using tools is a very big part of trying to be successful in this kind of area. So any advice you would give to undergrads in materials science or mechanical engineering and how they can kind of prepare themselves to kind of follow a career like this?
Okay. No, I think that's a great question. And I think my general advice would be that whether you're an undergrad or whether you're in grad school, students should be trying to develop that toolkit, and the toolkit that is going to enable them to solve the problems, whether it be at Adidas or some other company. Adidas as a company, culturally, what I have learned in working with them is that they're very sort of open-minded, they're very casual company. If you go to their company and their site in Portland, there's folks walking around in t-shirts and jeans, and they have a lot of open areas. So, they work in teams and they do a lot of discussion, and we spent a whole day just talking about the next generation of boosts and what the ideal cell structure would look like. So having a firm grasp, I think, of the fundamentals is key, because you might be working on graphite fiber for cleats. You might be working on fabrics that will be wicking your sweat. You might be working on using DIC to look at strains in the fabric or things like that. So having that sort of broad-based knowledge and really coming down to, as materials engineers and material scientists, we also always look at microstructure property relationships, and that's really what it comes down to. Tools, you mentioned tools. Tools to me are enablers to help us find the answer, and so I don't think that one should think about being, well, I am really good at tool A, B, or C. I'm good at understanding what that tool will get me to solve the problem. And that solving the problem is the most important thing.
Great. Okay, another question. What are the causes for the beads to delaminate at the boundaries?
So part of that is just incomplete fusion. So sometimes during the steam chesting process, again, you have beads that are not perfectly spherical. So they're elliptical to start with, and when you start to sort of apply pressure and steam, you're going to get some areas that maybe, it's also to go through that initial cell bed and how uniform is that steam, and it's able to come out as well. So, yeah.
So someone was saying, thanks for the excellent presentation. What is the reason for having the larger pore sizes on the inside and finer ones on the outside? I think this kind of relates to another question I had actually, was what if you kind of reverse the structure, pore it on the outside?
Exactly.
Yeah.
So I'll answer your question first because it's something that we started to look at in the last several months, and it's actually work in progress. So we were able to get beads that actually had that exact flipped structure. The reason why the original boost has finer cell sizes really has to do with the nature of the blowing agent. So the blowing agent comes in and it's actually not able to come out completely. There's a surface tension effect. And so that actually gives you the finer cell structure on the outside. But not only finer cell structure, but on the inside you have larger cells, but also thicker ligaments. So you have thicker, fatter ligaments in the inside. And so that also plays a role in what we found in the compressibility, the changes in mechanical properties.
I think that the animation you showed before, it was kind of like two nozzles where the beads are coming in. So if you were to have maybe multiple nozzles or going in, and so each bead doesn't have to travel as far,
right?
Then you could probably tune the structure that way too, right? But
exactly.
Yeah, the mold would be much more complicated, maybe not very.
Yeah, and because they're elliptical, in fact, some of the beads actually end up lying more flat. I mean, they're a little bit at an angle, but they do, sort of lie along the long direction.
Right. Okay, great. Does I think so. I think you answered this one. The beads already have this gradient structure, but is there a problem with during the blowing that it's being destroyed along the edge? Like if you kind of had too long of a blowing cycle or too rapid or too intense, maybe you would destroy completely that fine edge structure.
It has to be tailored, and I think you can get actually then fractures on the outside. And some of the things that they've also looked at is when they blow it too much, you get ligaments that are almost like very fine hairs. And we tested some of those, and those guys just collapsed, immediately, and they change in shape. You get a lot of permanent deformation, plastic deformation. So I think, and this original process was developed by BASF in collaboration with Adidas, and so I think they really did a nice job of tailoring not only the graded structure, but also a lot of that local buckling.
Very nice. Yeah. What are the smallest foam thicknesses and pore sizes possible for compression testing during tomography?
So in microtomography, we can do multiple cells because it's a function of density. So, with polymers, densities are relatively low. The resolution at the lab scale is about 1 micron, a little less than 1 micron. But we can get voxel sizes that are on the order of 200 nanometers. Okay, now I want to make a distinction, particularly for the students, that voxel size and resolution are not the same thing. So just because I have a voxel size that's 200 nanometers, I probably need to have 3 to 5 voxels to be able to really say that something is there and then I can see it. So, 700 nanometers, 800 nanometers, I would say at the lab scale is something we can do fairly routinely. There are other techniques if you have much smaller sizes, sample sizes, like at the Argonne National Laboratory, if anybody's interested, to do transmission X-ray microscopy, which is also tomography, and you can get down to 20 nanometer resolution. And for metals, you can typically do a volume that's maybe micron diameter by 20 micron in height kind of cylinders.
I think it goes back to another issue always that happens is you look smaller and smaller area, you can get more detail, but you lose, you're looking at one tree and trying to figure out what the forest is doing.
Exactly. And, and I think that's such a key point, Abdallah, because oftentimes we're trying to look finer and finer and finer, but we're losing track of sort of peripherally the bigger picture, right? It's almost like taking an SEM image at 100,000x without looking at the entire sample first.
Right. Okay. Yeah.
And so here at Purdue, we have a flat panel display where we can do, you know, tens of millimeters type samples. And then with the Zeiss system, we use what's called a scout and zoom technique, very technical term, where you scout an area and you can zoom in, and then you can take a higher resolution scan. So you could do multi-scale kind of things there too.
So I think you touched upon this already, but what are the next steps in the work?
So the next steps are really to look at novel kinds of you know, cellular structures, and there's a couple of other universities that are involved with actually processing some of the beads as well, with tomography and mechanical testing. One of the things that we are interested in looking at is now sort of, you know, think about two beads that are fused together, and if I could make a notch between those two beads and determine the interfacial toughness. So we've started to do some of those experiments in tomography as well. In other words, what kind of boundaries give you the best kind of toughness, and, you know, and also what is the effect of the base polymer in terms of affecting properties. So
I think that's all the questions from me. Anyone else have any questions? Maybe Jordan or Danielle?
I don't know if this, I don't exactly know how to word this question, but I know like a lot of the foam bases of all these Adidas shoes are kind of different shapes and obviously have different ridges on the bottom. So do those also play a factor in some of your toughness tests? And
yeah, that's something we'll need to look at, and there are different iterations of Boost. I think now the most recent one is Ultra Boost, which has a, you know, slightly different polymer composition, but very different, very similar graded structure. So I think what's key about Boost is really that graded structure, and so now, you know, there are other sort of processing challenges to try to make Boost and larger volume and faster rate of production and things like that.
Okay, yeah, if there's nothing else, thank you very much, Professor Nick. Great talk, very interesting. I think our members really enjoyed it. Thank you for being here and giving the time. And yeah, I think everyone else who attended, thank you for being here. Please look at asmontario.org for our Maze talk and then further beyond. We're going to keep trying to do these things to keep trying to get interesting speakers to you. And so please stick with us, and hopefully we'll be able to see you in person sometime in the near future.
Okay.
Thanks
very much. Thanks a lot.
Good day.
thank you for
Bye.
thank
Yeah, bye-bye.